Hello, this lecture is recorded for RCBC students, but we hope others will listen because science matters. We're going to be talking about Chapter 19, and in this lecture, it's going to be specifically about viruses. Future lectures will talk about bacteria and prions. Let's get a sense of what viruses are by looking at first at the size of the viruses. And you can see here on the left, we have a red blood cell. This human cell is 7 microns. That's a micrometer. And micrometers are 1,000,000 times smaller than a meter. So on average, we'll say that human cells, eukaryotic cells, are about 10 microns. Bacterial cells are much smaller. In this case, it looks like maybe it's only about 3 times smaller, but it can be 10 times smaller. And they have, of course, we already have studied these prokaryotes, have many less organelles and cell structures than we see in eukaryotic cells. Viruses are about 10 times smaller than a bacterial cell. They are not cells at all. As you can see here, we say they're particles, but the other two we described as being cells. We discussed early on that viruses are not considered living. They are parasitic. They need a host to infect, and they get the host and the host machinery to make copies of the virus for them. Let's take a look at a few different virus types. This is an adenovirus, which can be the cause of a common cold. It's very simple. We have this coat on the outside that we call a protein capsid. There are different types of proteins that can make up the capsid. In this depiction, we have one type here in green, and then this little spike is another protein. We also have a genome that is similar to our chromosomes, and their genome can be made up of RNA or DNA. That is unlike living things, where the chromosomes are always made up of DNA. We can add another layer of complexity in some viruses, such as smallpox. They not only have the genome, they have the protein capsid, but you can see here that they have a couple of envelopes. Those envelopes are made up of a phospholipid bilayer, just like our plasma membrane, but we don't call it a plasma membrane because that would indicate it is a cell. On that plasmid membrane, we also have, on that envelope, we also have proteins. And those proteins are going to become very important in getting into a cell. So if we look at this table here, we can see a comparison of viruses versus living things. And you can see that, as we mentioned, the hereditary material of viruses can be DNA or RNA. So their genes can be DNA or RNA. Both of them can either be single or double-stranded as well. And you know that in living things, it's always DNA that makes up the genes, and it's always double-stranded. Viruses do not have a plasma membrane. They have something called an envelope, but it is not a plasma membrane. Do they carry out transcription independently? Basically, no. They are going to utilize all the machinery of the host cell that they infect to carry out transcription and translation. We'll describe where they might bring a polymerase with them in a later lecture. And then metabolic capabilities, basically none. We take in food and nutrients from our environment, we break it down, we get ATP out of it. They don't do that, which is one of the things that defines something as living. They don't really need to do it because they're going to let us do all the work for them. Now, the viruses are typically two things: species specific and cell type specific or system specific, if you want to think about systems. A virus that is such as human immunodeficiency virus, for the most part, only infects humans, right? And that is because we will see there are specific proteins on the virus and specific proteins on humans that match up, and they match up, allowing the virus to get in. Also, those proteins specific to certain viruses are only found in certain areas throughout the body. So when we talk about COVID-19 and coronavirus, we know that that is related to the respiratory tract because the cells within the respiratory tract have the right proteins that allow the virus to get in. So let's talk a little bit about how viruses get in and what their life cycle is. And this is a very generic depiction. They are all very different in their, what we call their life cycle. We have here, this is an example of a retrovirus such as HIV. And in this retrovirus, we have, or on it, we have an envelope. That envelope has proteins in it, and inside it is an RNA virus. You can see that this is the cell, the plasma membrane of the host cell. You can see that the protein on the envelope interacts with the protein on the plasma membrane. That's going to allow the capsid and the RNA to get inside the cell. Once that happens, we get rid of those capsid proteins, and now the virus's RNA is free to do what it needs to do. In HIV, that RNA does a little bit of a trick to become DNA and gets inside the nucleus. Not all viruses work that way. Let's take a look at how the virus gets out. Once the virus can make a bunch of copies of its genome and all of the proteins that made up the envelope and the coat, what you can see is that they will begin to assemble together. They will bud off, and they will use the plasma membrane of the host to create their own envelope. God bless you. That was my cat. And the new viruses will then leave. In this case, they're budding off. They will leave the host cell. Now, it looked like one virus got in and maybe only a few got out, but it can be millions of viruses that get out of one cell, depending on the virus type. We're going to go into more detail, of course, looking at many different types of viruses, but the one that has all of our attention now is SARS Coronavirus 2. This stands for Severe Acute Respiratory Syndrome. Coronavirus, because these proteins here look like a crown. And it's number 2 because we also had a coronavirus, a SARS coronavirus, back in the early 2000s, but this one is a little bit different. If we take a look at this structure of the virus, it is made up of RNA, which is shown here. It does have an envelope, and on that envelope are proteins. This one here is hemagglutinin esterase, which you're going to hear that hemagglutinin again when we talk about influenza. And then this sort of signature spike glycoprotein, which is depicted here in yellow. And this glycoprotein, of course, is made up of amino acids, but there are sugars on it as well. This is the virus that has caused the pandemic that we are living in right now. So let's just briefly talk about the difference between what an epidemic is and a pandemic. An epidemic is a disease that rapidly infects a large number of individuals, and it tends to be more localized, whereas when it becomes worldwide or global epidemic, we call it a pandemic. Now, previously when I gave this lecture, the pandemic that I might discuss is the one from 100 years ago, which was called Spanish influenza. And we're going to talk about that, but we'll focus on the pandemic that we're living in right now. I would like to just mention that the thing that is particularly harmful about SARS coronavirus 2 is that it has a high infection rate and a high mortality rate. And that's what is the double-edged sword that makes this so dangerous. But I'd also like to say that we are trying to learn and understand about this pandemic as we are living through it. And that is a very difficult thing to do. So kudos to the people at the World Wide Health Organization and the CDC. It is sort of like, I was thinking about the analogy of going into, say, the Super Bowl, and you're playing somebody from Mars. You've never watched them play the game before. You don't know how to defend against them. And people want to know the score. They want to know the outcome. Tell me, are you going to beat this thing? You don't know it yet. And so it's very difficult because we're asking questions of the experts and they're learning about this at the same time. We don't have a lot of adequate tests available. So we're not even quite sure who has been infected with the virus or not. Perhaps many of us already have been and we've recovered. Maybe we have some antibodies. There's still a question as to whether we can be reinfected. And it's a really difficult place to be in. So we will pick up the next lecture with talking about what we do know about this virus, what its life cycle looks like, and the recommendations as they stand on April 17th, 2020. Thank you very much.